Welcome to G Fun Facts Online. Today, we are exploring a subject that sounds like it belongs in a fantasy novel, but is actually happening in laboratories right now. We are talking about the age of biological alchemy. For thousands of years, humans have chased the fountain of youth. Emperors and explorers sought elixirs to reverse the aging process, but it turns out the secret was never in a lost city or golden chalice. It was locked inside us all along. Deep within the nucleus of every cell in your body, there is a dormant instruction manual for immortality. We are going to discuss cellular reprogramming, a technology that is rewriting the code of life, curing incurable diseases, and challenging our very definition of what it means to be human. To understand why this is such a massive deal, we have to look at how biology used to view aging. For most of the 20th century, development was seen as a one-way street. Imagine a mountain. At the top of this mountain, you have a stem cell. It has infinite potential. As gravity pulls it down the mountain, it rolls into a valley and becomes a specific thing, like a heart cell, a skin cell, or a neuron. Once it settles in that valley, it is stuck. It cannot roll back up the hill. This concept was called the Waddington landscape. It meant that once a cell decided what it wanted to be, its identity was fixed, and its aging process was irreversible. A skin cell was a skin cell forever, until it died. But in 2006, a Japanese scientist named Shinya Yamanaka did something that broke biology. He effectively detonated a bomb at the base of that mountain. Working at Kyoto University, Yamanaka discovered that by tweaking just 4 specific genes, he could push the ball back up the hill. He took a mature, aging piece of skin from an adult mouse, applied his recipe, and turned it back into an embryonic state. He reset the biological clock to 0. These 4 genes, October 4, SOX2, KLF4, and CMYC, are now famous as the Yamanaka factors. The cells they create are called induced pluripotent stem cells, or iPSCs. They act exactly like embryonic stem cells, capable of becoming any tissue in the body, but they don't require an embryo. They are created from adult tissue. This discovery won Yamanaka the Nobel Prize in 2012, just 6 years after his breakthrough, which is unheard of in the slow-moving world of science. But how does it actually work? How do 4 proteins reverse time? Think of your DNA as a massive library. In a skin cell, the books on how to be skin are open and being read, while the books on how to be a neuron or an embryo are glued shut and buried in the back of the stacks. When the Yamanaka factors enter the cell, they act like librarians with bolt cutters. The factor CMYC acts as an amplifier, physically loosening the DNA structure so it can be accessed. Then, KLF4 and SOX2 hunt down the silenced stem cell genes, and October 4 turns them back on. Simultaneously, they scrub away the chemical tags, known as epigenetic marks, that told the cell it was old and mature. The cell literally forgets it was ever a piece of skin. Its metabolism shifts from the slow energy use of an adult to the rapid, sugar-hungry state of a growing embryo. However, this power comes with a dangerous price. This is the double-edged sword of cellular reprogramming. If you just inject these reprogrammed cells into a body without careful guidance, they don't know when to stop growing. They can form teratomas. These are chaotic, grotesque tumors that contain jumbled tissues like teeth, hair, bone, and muscle, all mixed together. Taming this wild potential, ensuring we get regeneration without cancer, has been the primary challenge of the last decade. But now, we are seeing the first wave of successes, moving from the petri dish to the patient. We are witnessing the first curative cell therapies. Let's look at Parkinson's disease. This condition is caused by the death of dopamine-producing neurons in the brain. It is a hardware failure. Drugs can help symptoms, but they cannot replace the missing cells. In a landmark trial initiated in China between 2024 and 2025, researchers did exactly that. They took skin or blood cells, reprogrammed them into stem cells, guided them to become dopamine neurons, and surgically injected them into patients' brains. Early reports describe a functional cure, with patients regaining motor control. In the United States, Blue Rock Therapeutics is doing similar work, aiming to graft new neurons that physically reintegrate into the brain's circuitry. Then there is the spinal cord. At Keio University in Japan, Professor Hideyuki Okano is pioneering a treatment where iPSC-derived neural stem cells are injected into spinal cord injuries. These cells don't just patch the hole. They differentiate into new neurons to bridge the severed connection and create support cells to re-insulate damaged nerves. In animal trials, paralyzed monkeys regain the ability to grasp objects. Now, human trials are underway to rebuild the central nervous system. We are also seeing incredible progress in restoring sight. The eye is a great target because it is less likely to reject foreign cells. Trials for age-related macular degeneration are using sheets of reprogrammed retinal cells to replace the dying layers in the back of the eye, effectively halting blindness and, in some cases, restoring pockets of vision. But here's where the story gets even bigger. If we can create young cells for transplant, could we simply reprogram the cells already inside us to make them young again? This question has launched the single biggest gold rush in biotech history. In 2022, a company called Altos Labs launched with $3 billion in funding, backed by figures like Jeff Bezos. Their goal is partial reprogramming. See, if you fully reprogram a cell, it loses its identity. You don't want your heart cells to forget they are heart cells and turn into stem cells. Your heart would stop beating. But scientists found a loophole. Reprogramming is a gradual process. The markers of age are erased in the first few days, but the markers of identity are erased later. By exposing cells to the Yamanaka factors for just a short burst, maybe 2 to 4 days, you can erase the accumulated damage of aging without erasing the cell's identity. The cell remains a skin cell, but it functions like a young one. This is based on the work of Dr. Steve Horvath, who discovered the epigenetic clock. We can now look at a cell's DNA methylation and predict an organism's age with terrified accuracy. Experiments by companies like Rejuvenate Bio have shown that partial reprogramming literally reverses this clock. In a study led by David Sinclair, researchers used a modified cocktail to restore vision in mice with glaucoma. They didn't just heal the injury, they reverted the neurons to a youthful state where they could regenerate on their own. This sector is exploding. You have Retro Biosciences, backed by Sam Altman of OpenAI, aiming to add 10 years to the healthy human lifespan using AI to design safer reprogramming factors. You have New Limit, focusing on restoring the immune system. The goal isn't just to live longer, but to stay biologically young. There is also a shortcut called direct reprogramming. Creating stem cells is slow, but what if you could skip the middleman? Researchers are learning to convert one adult cell type directly into another. After a heart attack, the heart heals with dead scar tissue, but by injecting specific factors, scientists can tell the scar tissue fibroblasts to transform directly into beating heart muscle cells. It is like alchemy, turning the lead of scar tissue into the gold of functional muscle right inside the patient's chest. However, as with any technology that touches the code of life, we are walking into an ethical minefield. Using iPSCs, scientists are now growing cerebral organoids, pea-sized, three-dimensional lumps of brain tissue. These mini-brains develop layers, fire electrical signals, and even synchronize waves similar to a developing fetus. As these organoids become more complex, potentially integrated with AI systems, we have to ask, can they suffer? If we grow a brain model from your skin cells to test a drug, and it develops a rudiment of consciousness, do we have the right to destroy it? Even more disruptive is in vitro gametogenesis, or IVG. Scientists have successfully turned mouse skin cells into eggs and sperm, creating viable baby mice from them. Applied to humans, this would mean infertility could be cured entirely. But it also means men could produce eggs, and women could produce sperm. A child could theoretically have genetic material from 4 people, or just 1. It opens the door to embryo farming, where clinics could generate thousands of embryos from skin cells and select the best one, ushering in a new era of eugenics. We have even seen the creation of synthetic human embryos that develop without any sperm or egg at all, challenging our legal and moral definitions of life. We are standing at the threshold of a new era. The convergence of artificial intelligence and cellular reprogramming is accelerating this progress exponentially. AI is now designing synthetic transcription factors that are 50 times more efficient than nature's originals. We are looking at a future of bioprinting organs on demand and personalized maintenance, where you might bank your cells at age 25 to print a new heart at age 70. Cellular reprogramming is not just a medical treatment, it is a philosophical earthquake. It tells us that aging is not an inevitable decline, but a buildup of errors that can be debugged. It tells us that our biological identity is fluid, not fixed. The alchemists of the past failed, but today's biologists have succeeded. The code has been cracked. Now we are just learning how to write the software. Thank you for listening to G Fun Facts Online. If you enjoyed this deep dive, stay tuned for our next episode.